Your lecturer is Professor David Sadova. Dr. Sadova is the Pritzker Family Foundation Professor of Biology Emeritus at Claremont McKenna, Pitzer, and Scripps, three of the Claremont colleges. He also is Adjunct Professor of Cancer Cell Biology at the City of Hope Medical Center. At Claremont, he has twice won the Huntoon Award for Superior Teaching. Dr. Sadova is the author or co-author of more than 55 peer-reviewed scientific research papers and 5 books, including 2 recent biology textbooks, Life, the Science of Biology, and Principles of Life. A laboratory scientist, Dr. Sadova's research concerns resistance to chemotherapy in human lung cancer, with a view to developing new plant-based medicines to treat this disease. He received his Ph.D. in biology from the University of California, San Diego.
Malignant, potentially deadly cancer affects 1 person in 3 in North America, Europe, and Australia, and kills 1 person in 4. My name is David Sadova, and my aim is to tell you about the science that underlies cancer. I'm going to tell you amazing stories about how cancer works to subvert the body's normal functioning, and hopefully, with our understanding, how we can treat it. This is Kareem Abdul-Jabbar during his career in the National Basketball Association. Kareem, for those of you who don't know him or don't know of him, for those who are not basketball fans, he was one of the most talented basketball players in the 20th century. He led his high school team in New York City to 3 national championships, at UCLA, his college, to 3 national championships, and with Milwaukee and Los Angeles and the National Basketball Association to 6 championships, was 6 times most valuable player, etc. Everywhere he played, he set records, most of which still stand. He retired in 1989. But all of his athletic ability didn't insulate him from cancer. Late in 2008, he developed symptoms of hot flashes and night sweats, and a medical exam added fullness in his left side, which indicated an enlarged spleen. When his blood was examined, the doctors had a diagnosis. In normal blood, typically, there are 5,000 white blood cells in a milliliter, about a teaspoonful of blood. In his case, there were 200,000 per milliliter. The diagnosis was chronic myelogenous leukemia. He had many of these blast cells that crowd out red blood cells. And when they do that, these leukemia cells prevent red blood cells from carrying oxygen, etc., and so people will have anemia. They crowd out the platelets, the blood clotting cells, and so people will have bleeding as well. They crowd out white blood cells involved in immunity, and so people have infections. Now, he had a moderate number of blast cells, so he was called the chronic phase. The name is not that important. But let's think of what would have happened if he'd gotten chronic myelogenous leukemia in 1998 instead of 2008, 10 years previously. The treatment of leukemia, like the treatment of most cancers up until the 21st century, was to treat dividing cells. You know that cancer involves normal cells going wild and reproducing too rapidly, so they give them drugs to stop the cancer cells from reproducing. Sometimes the dose of the drugs was not effective in leukemia treatment, and so they would use something called bone marrow transplantation or stem cell transplantation to improve survival. The spleen enlarges at the late stages, so the doctors would do surgery to remove the spleen. And as a result of these 3 approaches, survival improved to about 10 years from normally 2 years. But in the case of this basketball player, Abdul-Jabbar, this retired basketball player, he's probably going to have a normal lifespan. How did it all happen? This happened because of a triumph of science. And the triumph of science is a deeper understanding of what's going on with disease, and in this case, cancer. In conventional medicine, the idea is to see what happened and to try to treat the symptoms. So, for example, in conventional medicine for a leukemia, you will find out what's wrong at the cell level and treat the symptoms, which are dividing cells. Give a drug that will stop cells from dividing. And you know intuitively that's going to stop every other cell from dividing in the body as well. In molecular medicine, the new medicine as applied to coming from molecular biology, the idea is to find out exactly what's wrong deep inside the cell at the chemical level and then cure the disease. This is called targeted therapy. And this is what happened with Abdul-Jabbar. If we look deep inside the leukemia cell, we will discover, not surprisingly, that the cell is controlled, its function, by DNA. DNA is a chemical, it's an information system, and it's inherited. Every cell in the body has 6 feet worth of DNA. I want you to just consider what I just said. 6 feet of DNA, that's my height, of DNA crammed into something you can't see. There's 60 trillion cells in the body. So it's a lot of scrunching going on. DNA encodes cell functions, and the cell functions are mainly expressed as proteins. So these are the proteins that essentially are the functional aspect of DNA. It's like an architect. An architect has plans on paper, and the actual building is something else entirely. All cells have the entire inherited DNA, but they make only certain proteins. So for example, hair follicle, I have my hair here, makes keratin, the protein of hair. A red blood cell makes hemoglobin, which you may be familiar with as the red pigment. DNA is packaged, as I mentioned, kind of like spaghetti. It is packaged into smaller, scrunched up things called chromosomes. It has to be packaged if you've got 6 feet of DNA in a cell that's so small you can't see it. The chromosomes in humans happen to be in 23 volumes. That is, there are 23 volumes in our genetic encyclopedia, and it's like the letter A and the letter B and the letter C, just different articles. We inherit 1 volume of each chromosome from our parents, 1 from mother, 1 from father. And when you look at human chromosomes under a microscope, you see that here are the chromosomes all scattered around. In this instance, they are stained with different colors. In the 1970s, methods were developed to stain chromosomes specifically, and each chromosome pair is stained a different color. So for example, chromosome 1 is yellow, chromosome 2 is red, etc. This method allowed people to look at highly specific and subtle chromosome changes, missing pieces or pieces that have been swapped. And in the case of the chronic myelogenous leukemia that Abdul-Jabbar has, there was a swap of 1 piece of a chromosome for another. Here is chromosome 9, and here is chromosome 22. And in the chronic myelogenous leukemia cell, and nowhere else in the body, at no other time in the life of a person, there is a swap that happens. A piece at the end of chromosome 9 is exchanged and swapped for a piece at the end of chromosome 22. Now, just as in an encyclopedia of articles, we have genes. And these are the articles at the end of chromosome 9. It's called ABL. The exact nature of it is not important for us at this time. And the one at the end of chromosome 22 is BCR. And we cut them in 2, and we do this swap. And at the end of chromosome 22, we have a very small piece here at the end that is half BCR from chromosome 22 and half ABL from chromosome 9. This is called a fusion gene. And as I mentioned, DNA is expressed as proteins, and so this gene will be expressed as an abnormal protein. The fusion protein is what stimulates cell division. These findings that I just described use new ways to study life, called molecular biology. And the new protein was first purified. It was made in a laboratory. A drug then was made against that protein specifically, and that drug hopefully would block its function, which normally is to stimulate cell division. The drug is called Gleevec, and it was into clinical trials pretty quickly in the late 1990s into 2000. And here is the research article coming from a very high prestige journal, the New England Journal of Medicine. And you can even read the title, Efficacy and Safety, you know what those are, of a specific inhibitor, that's what we're blocking, of BCR-ABL, that's the protein that we're interested in. Tyrosine kinase, don't worry about right now, in chronic myelogenous leukemia. And if you look at the authors, it's a whole big team. The era of a single scientist working alone is over. Science is now done in teams. And here, the interdisciplinary team included doctors who coordinated the clinical trial, doctors who were treating the patients, statisticians to analyze the data, chemists to design the drug, nurses to take care of the patients, etc. It's an amazing process, and the coordination was done by Brian Drucker, here in this case, at Oregon Health Sciences University. Before we look at the results of the Gleevec clinical trial, I want to introduce a concept to you that we're going to see throughout the course, and that is the Kaplan-Meier survival curve. This is a curve that looks at the progress of people who are surviving, or depending what the endpoint is, getting better, whatever, against time. So on the y-axis, you have survival, and it starts at 100% or 1.0, and it ends up, in the case where we're looking at survival, with 0, which means everyone is deceased, and years of diagnosis. So this is a good survival curve. It's going down very slowly. People die a natural death. This is a poor prognosis survival curve. Now let's take a look at what happened with Gleevec and the new therapies for chronic myelogenous leukemia. It was an amazing transition, as I've mentioned. In the 1960s, survival was not very good at all. Again, here's surviving and years of referral, which would be years after diagnosis. Then in the 1970s, it got better. In the 1980s, it got even better. In the 1990s, as I mentioned, is about 10 years, right, doing a lot better. And in the 2000s, essentially survival is normal lifespan for people. It's just an amazing story and a triumph of what is called molecular medicine. This is going to be one of the themes of this course, how science has been applied to the cancer problem. In this lecture, and the one that follows, I'm going to discuss how cancer is an important challenge to humanity. First, I'll describe some aspects of the history of cancer, and you'll see how it really reflects the history of medicine in general. Then I'll talk about a war against cancer you may have heard about, and then I'll briefly outline the course. In the next lecture, I'll describe how cancer is a burden on society, both at an individual and, sadly, at economic level. Then how cancer increased as a medical problem in the past century, and finally, cancer as a global challenge. So we begin by describing cancer history. Now the question is, how do you look at the history of anything? Well, one thing I've learned from great courses that I have heard, and I've seen a lot of great courses myself, I'm a customer as well as a lecturer, is that history can improve our understanding of the present. And for cancer, history may improve our concepts of the causes of cancer, the treatment, and the prevention. Cancer history is actually studied in two ways. First, by actual specimens, and second, by written materials. First, specimens. Well, there are two types of specimens we can study. First are fossil bones, because the soft parts of the body usually are not preserved over time. And secondly, we can look at mummies. First, bones. If you look at this skull that's about 2,500 years old, you see that there are holes in the top of the skull. And the symmetry of those holes indicates that there must be some cancer that was there that spread to these bones. What's amazing about this particular skull, which comes from eastern Slovakia, is that the cancer has been identified because people extracted some chemicals that were still around the skull and found a marker for prostate cancer. So amazingly, this is the skull of a man who had prostate cancer that metastasized or spread to the skull. Mummies. Well, you can do, is a mummy going into a CT scanner. You can look at mummies and look at cancers, and people have found large tumors in mummies in ancient Egypt about 2,000 years ago. But otherwise, cancer in these remains, in skulls and in mummies, etc., is pretty rare. People in days gone by generally didn't live very long. And as we're going to find out, cancer is a disease of old people. Now, the second way we can study cancer is by written materials. There are oracles handed down, dated from 1000 BC in China, associating cancer with diet. But when you look through history at all of the writings about cancer, we come up with about 3 ideas for the cause of cancer. And those 3 ideas are, first of all, it's a punishment by gods, a religious idea. Secondly, that cancer is caused by an imbalance of some chemicals in the body or something in the body called humors. And third, the idea that cancer is a disease of cells and chemicals inside of cells. First, the idea of cancer as a religious punishment, for example. This is the Ebers Papyrus. It was discovered in Luxor, Egypt. It's dated at 1538 BC. And if this is translated into English, it says, describing a cancer, and it says, cancer is a growth against the god Zenus, and the advice is, do thou nothing thereagainst. I'm going to repeat that. Do thou nothing thereagainst. In other words, we don't want to offend the god, we'll just let people progress with the disease and probably die. Another papyrus in ancient Egypt describes 8 cases of breast cancer. And again, do thou nothing, just use palliative care. So that's the first idea people have had for cancer. It's a punishment by God or gods. Does it still exist in our society? Of course it does. You probably know people who feel deep inside of them, if they get cancer, that they've done something morally wrong or there's some problem here. There's a punishment that God is doing to them. And that's an idea of disease that many people have. Now, the second idea I mentioned is the idea of humors. Here are 2 famous doctors of the ancient world. One is Hippocrates, Greek, and the other is Galen, a Greco-Roman person. Hippocrates studied cancer very carefully. He coined the term cancer karkinos, which is the Greek word for crab, because some tumors had these hard shells and projections coming out. They looked like a crab. Greek medicine believed, and Hippocrates believed, that the body was controlled by 4 fluids. He called them blood, phlegm, yellow bile, and black bile. Blood, phlegm, yellow bile, and black bile. And that cancer, he proposed, was caused by an excess of black bile. Galen, the Greco-Roman physician, living a couple hundred years later, 130 to 200 AD, translated the word karkinos into Latin for cancer and gave us the word onkos for mass, which is now oncology. Galen also believed in an imbalance of humors and also the idea of ancient Egyptians, you can't do anything about it, just leave it there. And this idea of an imbalance of humors, black bile being in excess, and the do-nothing idea held sway for 1,500 years in Western culture. So that's the second idea of a cause, an imbalance of humors. Does that persist today? Yes, it does. And part of my research, I've actually collaborated with traditional Chinese medicine doctors. And in traditional Chinese medicine, they try to study a humor that is something we can't define chemically, but it's an essence called the qi. You've heard of Qi Kung, you've heard of Tai Chi. The qi is the life force that Chinese medicine manipulates. Again, it's very much along the lines of humors. The third idea, that is, a cancer is a cellular disease with chemicals, really begins in the Renaissance in the 15th to 17th century Europe. Leonardo da Vinci, other people, did autopsies for looking at causes of death. And they really invented the modern era of pathology, where you looked at organs and what was going on. This led to advances in surgery to remove tumors. This is a famous portrait in art of a cancer surgery that is going on by a Dutch painter. In 1770, a Scottish surgeon named John Hunter said that if a tumor is localized, he said, I'm going to quote this, no impropriety in removing it. There's no impropriety in removing it. That's totally against what Galen and Hippocrates had said, which was do now nothing, right? Now they said, oh, we can take them out. And by the 19th century, with advances in anesthesia and antiseptics, more people survived surgery. In addition, the modern era of biological science came in the 19th and 20th centuries. Now, biological science really has 3 major ideas. And those 3 ideas are mechanism, the cell theory, and the third one is evolution by natural selection. First, mechanism. Mechanism in life science says that cancer, like all life, has a chemical physical basis. They're not humors that are balancing, etc., but they're chemicals that we can identify. Cell theory says that cancer is a disease of cells, those building blocks of life, 60 trillion of them in humans, and those cells divide and cause a tumor. And this can be related, of course, to the Gleevec story of attacking something going on inside cells. Evolution applies to cancer because, as you will see, as tumors grow and develop, they change. Mutations happen, the tumor gets more difficult to treat. The cancer that ends up growing very large is very different sometimes than the cancer that began. So that's the third idea, cancer as a mechanistic cellular disease, and that persists today with most scientists and physicians. In this lecture and the one to follow, I'm going to talk about cancer as an important challenge to humanity. I'm going to look at the history of cancer a little bit, which I've done. I'm going to talk about war against cancer at this point. And then I'll describe how the science of cancer is going to be dealt with in this course. In 1971, on December 23rd, Richard Nixon, then President of the United States, signed the National Cancer Act to coordinate U.S. efforts. He declared war on cancer with actually a stated aim to eliminate the disease in the decade of the 1970s. Well, I have two comments, of course. The first comment is obviously it didn't work because we still have cancer with us. And my second comment is, well, why did it, it's a question, why did it happen then? Why did we come to 1971 with this great optimism that we could eliminate cancer? There are really two events that happened in the 20th century with cancer that led to this optimism. The first is the invention of cancer chemotherapy and its effectiveness, drugs to treat cancer. And the second is the idea of cancer viruses. The first 60 years of the 20th century in life science was the era of biochemistry. That is, looking at all the little chemicals inside the cell and seeing how they were transformed and what they did, and how they were controlled by proteins. In this case, each chemical is catalyzed by an enzyme, usually a protein, encoded for by DNA. So the idea that DNA is necessary for cancer growth and that proteins and chemical changes happen led to three major advances in chemotherapy up until 1970. I'm going to describe only three people. There are many other people one could describe. First, Charles Huggins, in 1939, won the Nobel Prize. He was a Canadian, got his MD at Harvard, and his career at the University of Chicago. He proposed that prostate cancer is stimulated to grow by a hormone, the male sex hormone testosterone, and said, well, the way we can treat it is by eliminating testosterone. Now, there are two ways to do that: the surgical way, which would be castration, and the second way was to design an anti-cancer drug, an anti-testosterone drug, which he did. And this anti-testosterone drug alleviated the symptoms and the growth of prostate cancer. It worked. And that really began, before World War II, the era of modern chemotherapy. In the 1940s, Sidney Farber, working at Harvard Medical School, found that a vitamin named folic acid was necessary for bone marrow cell production. And at the chemical level, they found the biochemical pathway involved where folate was needed. And so, reasoning from that, he said, well, if it's necessary for bone marrow to make white blood cells, if I have an anti-folate, something that will block folic acid, perhaps that will be able to treat leukemia. The drug is called methotrexate. It was very successful with children. In the 1950s, two scientists who we always say together, George Hitchings and Gertrude Ilian, they also won the Nobel Prize, working at a drug company, realizing that DNA is necessary for cancer because DNA has to be reproduced for cells to divide, made anti-DNA molecules, the first anti-DNA molecules. And these were successful as well. So those are the three major advances in chemotherapy. Now I'll add a fourth, which was in the 1960s. Scientists realized that cancers are not homogeneous. All the cells of a leukemia might not be the same. And so for some cancers, they developed combination chemotherapy of different drugs. We'll talk about this later in the course. And in this case, for Hodgkin's disease in children, and I know people my age who had Hodgkin's disease, it was normally lethal. They got combination chemotherapy, and they were cured, and they're still alive. So there's great optimism with these new drugs. The second idea for cancer was that it's an infectious disease. In the 19th and 20th century, there was a great development in medicine called the germ theory by Louis Pasteur, Robert Koch, and other great microbiologists. And they found that diseases, scourges of humanity, like tuberculosis and smallpox, etc., were caused by things like bacteria and viruses, and that we could treat them using antibiotics or even prevent it using vaccines in some cases. This idea reached its peak in 1926 when the Nobel Prize Committee gave the award to Johannes Fiebiger in Copenhagen, who said, I found growths, which he called cancer, in the stomachs of wild rats that were infected with a worm. He even named the worm Spiropteroba carcinoma, right? The name of a worm was called cancer-causing. And the Nobel Award said that he had succeeded in lifting with a sure hand a corner of the veil which hid us from the etiology of the disease. There's a huge but to this story, namely that it wasn't cancer, it was just an inflammation. He reported no data. No one could repeat it as well. So the story kind of died, and the Nobel Committee has been rather careful ever since. In 1910, actually, a virus was discovered that caused cancer in chickens, and by the 1960s, there were many tumor viruses in animals that had been identified, especially in rodents. So now we're leading up to this National Cancer Act, and some of these tumor viruses are an unusual way to reproduce. RNA was used rather than DNA. We'll talk about this later on. And Nobel Prizes were given out. There was great excitement that cancer might be a virus cause. Imagine, in that case, then you could treat it with a vaccine or prevention. This led a woman named Mary Lasker in the late 1960s, who was an activist against cancer. Her husband was an advertising executive who had died of cancer. She used all the skills of PR to lobby for the National Cancer Act, and there it was passed. Meanwhile, in Europe, there was a World Cancer Association that was passed, was founded, and there's a World Cancer Day every year on February the 4th. So great optimism in the 1970s, but we really haven't made much of a dent. Still, 1 in 4 people in our societies die of cancer. In this course, I'm going to describe cancer in terms of science. And I'm going to do it under a number of general headings. The challenge of cancer is the first heading. And that is, I'm going to describe, and I've begun in this lecture, but in subsequent lectures, the burden of disease in human populations with regard to cancer, and trends that are happening with regard to the increase in cancer, and what may happen for these trends in the future. In the second part of the course, I'm going to describe the causes of cancer. Now, how can we identify whether something causes cancer or how it causes cancer? There are really 2 ways to do it. The first way is a science called epidemiology, which is the science of studying diseases in populations. And I'll describe what that science is and what types of results it can get. But you know, epidemiology, looking at a large group of people, doesn't really prove something. It proves a very strong relationship between some outside agent like radiation and cancer, but it doesn't prove it. And then so you need to do experiments as well and try to find the exact mechanism, and that is the field of carcinogenesis. Things that cause cancer are called carcinogens. We will also learn in this part of the course that there are spontaneous changes that happen that we can do nothing about just because we're made of chemicals. And those spontaneous changes in DNA and other systems of the body will mean that cancer will always be with us because those changes will always happen, even if there are no carcinogens from the outside. The third aspect of the course will deal with cancer as a disease. I mentioned it's a cellular disease caused by changes in cells and some chemicals inside the cells, but the properties of cells. And I'll try to answer questions about how does a tumor grow? How rapidly does it grow? How does a tumor cause disease? That is, a tumor just might be a ball of cells that keeps growing. Why does it cause disease in the body? How does a tumor get identified? We'll see what the identification of tumors. How is diagnosis done of a tumor as it grows? How does a tumor spread? The most feared aspect of cancer is tumor spread or metastasis in the body. Finally, how does a tumor recruit its own blood supply? And this has become a very important topic in the last 10 years of cancer research, as we'll see. So with this background of causes of cancer and cancer as a disease, we'll go to part 4, where I will present the most recent information that we have on cancer, which is discussing genes, viruses, and the molecular biology of cancer. This is peering inside the cancer cell at its deepest level. And we'll see how some viruses do cause cancer, others cause just a modest increase of cell number. But from the discovery of cancer viruses came the identification of genetic material normally present inside the cell, which does the same thing that cancer viruses do, and that is stimulate cells to divide. I'll also describe under this heading inherited cancer, and how looking at the molecular biology of inherited cancer at the DNA level, at the protein level, led to the idea of genes that stop cells from dividing normally instead of stimulating them, and how those genes go wrong, thereby allowing cells to reproduce. I'll give the analogy of a tumor growth being just like a car, where you have to put on the gas pedal and release the brake. And there are normally gas pedals and brakes analogies in human cancers. This is the newest part of cancer research. In part 5, I'll describe cancer treatment. The major forms of cancer treatment are surgery, radiation, and chemotherapy. By far the most important and common one, by the way, is surgery. Most cancer is treated in some way with surgery. And surgery is the most likely one to be curative. I'll describe how these have evolved over time and changed in the last century to respond to scientific investigations. They don't come just out of thin air. they come from rigorous science, and these changes have happened because of rigorous science. In the last part of the course, I'll circle back to where we started with populations and talk about cancer screening and prevention and see whether those can do any good about the incidence of cancer in our population. In this lecture, I've introduced the history of cancer, reflecting the history of medical science. I've talked about the war on cancer and what's happened and what led up to that war and what's happened since in general. And finally, I've introduced you to the rest of the course. Thank you very much.